All right, so welcome to today's program titled Mass Spectrometry-based Proteomics in Biological Research. And before we begin, I'd just like to acknowledge the traditional custodians on the lands on which we meet today. And we're coming from all over the place, so we're meeting on many lands. I believe we have people from overseas. So welcome to everybody, and I hope you enjoyed today's session. So now that we've passed through the introduction, and hopefully with a few more people joining us online, we'll begin the program for today. And so we've got two speakers. We've got Dr. Rohan Lowe, who's going to be talking about our proteomics platform, but more about the theoretical concepts of proteomics. And then we've got Dr. Keshava Datta, who will talk about more the applications of proteomics and introduce that to you. So I'm going to stop sharing my screen and hand over to Rohan.
I'm going to take you through some of the basics. This is very much a whirlwind tour of LC-MS proteomics. There's only so much we can do in a short amount of time, but the idea is just to sort of almost fire your imagination or fill in a few little gaps that we can talk about in the question sessions after each short presentation, and then also later learning. Maybe you can come back to us and talk later. So there's really four areas I want to cover in this short presentation around sample preparation, preparing the proteins for proteomics, liquid chromatography, because we at the platform use LC-MS based proteomics. Chromatography is a very important part of that. Next, often the rock star of the process is the mass spec instrument itself. And then finally wrapping up with, there's a bit of computation required to actually analyze mass spectra and convert that information into proteins. So I'll go through each of these in detail as we go through. But just to start off with, what is proteomics? Really, in a very basic level, proteomics is the act of characterizing all proteins in a particular sample. It's the proteome. Proteomics is the art of studying the proteome. And really what we're interested in basically is to determine presence. Is a protein there or is it not? Is that protein modified in any way? You might be interested in whether it's phosphorylated or not phosphorylated. And the other question is, of course, is that protein's abundance changing over time? Maybe, or in response to, say, a drug treatment. So that's really three basic things that we deal with when we talk about proteomics and the study of these proteins. But there's a lot of problems associated with that. And the main one is complexity. And we'll come back to this again and again as we go through today. A biological sample contains thousands and thousands of different proteins. And the whole thing is just too complex to analyze with a mass spec, for example, in one hit. So we basically deploy multiple different techniques and technologies. We bring them all together, and that them acting in concert means that we can resolve and identify individual proteins in a complex mixture. So let's talk about some of those problems and the complexity around doing LC-MS proteomics. I mentioned initially, the slide's a little bit busy, I'll apologize for that. But basically, we think about different stages. Initially, you've got to go from a cell and you must extract protein out of that cell. So we have to lyse the tissue, we have to selectively extract proteins out of it and try and get rid of the nucleic acids, get rid of lipid, carbohydrates and things we don't want, so that it's nice and uniform. Then we have to deal with the problem where proteins have different sizes and are highly diverse. So what we do there is we digest our protein into peptides, and we instead deal with peptides in our experiments because they have much more similar characteristics, simpler, it's an easier to find question to solve. I mentioned liquid chromatography. We use liquid chromatography to deal with that complexity to analyze all these peptides at once. So instead, we use separation science to split them up and allow the mass spec more time to analyze each peptide, rather than having to do thousands and thousands in one hit, we spread it out over an hour or so and give it more time. And then, of course, the mass spec is there because we have to measure the mass of peptides and also fragment them and break them up and see what they're made of to determine what that amino acid sequence is. And that's because different peptides have similar physical characteristics and we must smash them up. The last step, that computational step, the data analysis step, is because it's simply too complex to analyze thousands of spectra manually. You can do it for one, but multiple spectra are too hard. So we use computation and statistics to sift through all those spectra and identify peptides in a sample. I've got a few slides just covering this process again to help you sort of fix it in your mind. At the beginning, we start out with cells and tissue, and we extract protein from that. So that's an important step of getting nice, clean protein for doing proteomics. Then, to reduce that complexity and make it easy for the mass spec to deal with, we digest it using, say, an enzyme like trypsin, and we break up a large protein into small peptides, more uniform characteristics, mass spec friendly. We then use liquid chromatography to separate a whole bunch of peptides into a less complex sample, and we introduce it to the mass spec using an electrospray source. And then the mass spec's job is to measure the mass of these peptides and fragmenting them in repeated cycles constantly for an hour or 2 hours per sample and collect the mass spectra. And then we move to the data analysis side, where once we have a series of mass spectra in a file, we can pass those on to some software, which will then compare the spectra to a theoretical proteome and decide what was actually present and what wasn't. So I'm going to go through again these steps repeatedly, and hopefully they'll start to stick a little in your mind on these basic processes and the concepts that are going on when we do LC-MS proteomics. So we're going to go back to that sample preparation, back to the start. So I mentioned we need to have our extracted protein, and we need to convert it into these peptides that are friendly for the mass spec. So what does that involve? Well, there are three basic processes that occur for that. First one is to denature the protein, and that's to open it up. It's normally folded if it's in solution. We want to open it up and allow the enzymes that we're going to digest it with access to that polypeptide. So we denature it, and typically you could use something like urea or a strong detergent to denature your protein. Then the next step is most proteins have one or more disulfide bonds between cysteines. These are a problem for the mass spec because it increases complexity. We must remove them. So we use a reducing agent like TSEP, and that reducing agent will break those bonds, and now we have a nice linear polypeptide. And we can then cap those free thiols to stop them sticking back together again using an alkylating reagent like iodoacetamide. And that keeps them nice and safe and happy. And then once we've done that step, we then digest it using something like trypsin to break up those that long polypeptide into nice little relatively uniform segments that are nice and easy for the mass spec to deal with. So that was the step one, the sample prep. Now we've moved out of the lab, and we're going to inject our sample onto an instrument and do the liquid chromatography stage. And so this is all about reducing complexity of thousands and thousands of different peptides into something that the mass spec can manage in a nice orderly fashion. So it's like taking a big pile of boxes in a factory and sifting them and sorting them into a nice linear production line type scenario, so that in our case, the mass spec is on the end of this production line. It means it can deal with them in a nice orderly fashion and a reproducible fashion as well. So this gives the mass spec time to analyze each peptide. Simpler spectra are beneficial for the data analysis stage as well. And also the liquid chromatography allows us to separate peptides that have the same mass but different sequences. So the mass spec couldn't actually tell them apart just by mass, but the LC stage may actually separate isomers out, so we could distinguish them by retention time, which is quite handy. So here's a picture on the right of our UHPLC system. It's very fancy, very high performance for this. Here's a picture of our column that we use to separate our peptides before they go into the mass spec. And we use reverse phase chromatography with a C18 functional group. It's non-polar, and the idea is that we bind the peptides to the column and then slowly, gradually elute them off using a gradient of solvent flowing through the column, which gradually changes from polar to non-polar as we increase the amount of a solvent such as acetonitrile is very commonly used. We also have a bit of acid in our solvent to encourage ionization of peptides when they enter the mass spec at the end of this process. And this is quite unusual HPLC system in that we run incredibly low flow rates, less than a microliter every minute, and at very high pressure, many thousands of PSI. So it's very high performance equipment. After that stage, we then move on to the mass spec. So the mass spec is where we measure the mass of those peptides and break them open and determine what amino acids each peptide has. So the first stage that's really important is we have to go from a liquid chromatography stage into a mass spec, and it must be converted from liquid to gas phase. And this is we use an electrospray ionization source, which is basically a very small nozzle with a lot of voltage on it. It sprays the liquid evaporates and leaves behind charged peptides in a gas phase. And that's what the mass spec can analyze. Mass spec will measure, actually, it does a mass measurement, but it's mass divided by charge. And this is an important concept to keep in your mind, that usually we talk about M over Z, MZ, and this is mass divided by the charge. So if a molecule has a lot of charges, the MZ becomes lower for a given mass. Something else that's important to keep in mind is that the whole process of LC and MS is multidimensional because we have time. We might do a LC run for an hour or 2 hours, and all that time we're collecting mass measurements which have mass and abundance. So we get this multidimensional type of data. And there's a diagram up in the top right here which explains one of the concepts of these sort of spectra are like a slice through this graph of abundance over time. And each slice shown in purple here has a mass spectra with different M over Z values and their abundances. So you have these multiple slices occurring by the mass spec over time as it's taking these snapshots of mass spectra and processing those spectra. So the other important thing to keep in mind with LC-MS proteomics is the mass spec is doing what we call MS-MS or tandem mass spec, where there's multiple measurements occurring for each peptide as it comes through the mass spec. And it's these cycles of mass measurement and fragmentation and measurement again. So let's have a look where we're going to start here. I think on the diagram on the left, we've got an initial scan, which we call MS1, and then a particular peptide that was found in that scan will get picked and it will get smashed up by fragmentation, and then another scan will occur where we measure the size and what fragments were produced from that original peptide. So we get these cycles of MS1 and MS2 scans. And you can do multiple selection and fragmentation and measurement before we go back and collect another big MS1 scan. So we have a look on the panel more on the right in the red box. the selection phase, you might have a whole bunch of peptides come into the mass spec at once. You get a lot of peaks on your mass spectra, different NZ values with abundances, and the mass spec will pick one of them, which I put a star on here. It'll pick just one of them and it'll say, okay, let's find out what that's made of. And it will pick that one, isolate it from all the rest, smash it up, and then measure the size of those fragments and say, okay, now I know what the starting mass was. I know what all the pieces mass were. And that it's actually done its job at that point, and the software later on will try and reassemble that jigsaw puzzle, so to speak. But the mass spec is really trying to just tick these boxes. It's trying to measure all of the ions that are coming into it, which in our case should be all peptides, and it's trying to smash them up and measure the mass of all those fragments. And it does that repeatedly over and over again with cycles of MS1 and MS2 scans, repeated fragmentation of different ions until it's fragmented everything it can see, and then it will go back and collect a new scan again at the start. And this way, these repeated slices will in theory collect all. Generally it won't, but everything's slightly imperfect. But the idea is that way it's constantly scanning, fragmenting, picking, fragmenting through the whole HPLC run over that 1 or 2 hour process. And it's going to collect a lot of spectra during that process. In our experiments, typically we would collect somewhere around 100,000 spectra for a 2 hour run. So the final stage is the computational part, which is a database search. So what we've got at that stage is we've taken our protein, we fragmented it, we digested it into peptides. We separated the peptides on the LC. We measured a whole bunch of spectra from those peptides using the mass spec. Now we use a computer to search a database of known proteins for that organism and match spectra to theoretical values for that proteome. And if they match up perfectly, we get a tick and we say that peptide is totally part of this rabbit proteome or a Arabidopsis proteome or whatever it is we're looking at, and we'll record it. So some of the important concepts for the database search is that the software only really finds what we tell it to look for. So it's very important to configure it correctly with the right parameters to start off with. And typically that means things like if we modified something like alkylating those cysteines, we have to tell it that. Say, okay, look out for alkylated cysteines. The mass will be slightly different than just a plain cysteine. We have to tell the software what enzyme we use to break up the peptides. Those have specific sequences that they cut at. So if the software knows that, it will follow those rules. And if we use a different enzyme, it's not going to work at all because the computer's looking for a different cut point than what happened in reality. For the sequence database, we must match the database to the correct species. No point using a plant database to match mammalian peptides to. That won't work. So we get the right species, we specify the right contaminant proteins. If we think, oh, there's going to be a lot of Chinese hamster cells getting through into my express protein, I need to put the Chinese hamster database in there as well. We've got to set our statistical thresholds correctly so that we don't get too many false positive hits coming through. And if we introduce any fancy labels in a more advanced technique to our protein right at the beginning or during that sample prep stage, we must tell the database search that we did that. So did we add a biotin tag? Did we cross-link something? Did we add some fancy label? Again, the database search must have all of the correct parameters for the reality of that sample. Some other points here I wanted to make about the database search, I think I've got a few more minutes, is we can do some what's called label-free quantitation of protein abundance using software that can calculate that amounts to determine if a protein is changing abundance over time. but it's best to compare a single protein across samples because basically it's not a particularly inherently quantitative process. And what that means is that, yes, you definitely know if a protein is changing abundance over time or in different samples, but it's hard to know which protein is more abundant than another because each protein will behave slightly different in the process. And the other thing to keep in mind with your database search when doing LC-MS proteomics is the results are somewhat software dependent, not entirely, but there's that 5% difference, which tends to be brought in by which software you use to do the search. Some, they'll play to their own strengths and weaknesses. And just keep in mind that if you use a different search, you will get a slightly different outcome because the software does make little decisions and have particular efficiencies. MaxQuant, or with its Andromeda search engine, is quite popular. It's a free open source academic engine. And you can even try something that's called Search GUI, which can combine multiple different searches together to try and reduce that bias. So that's basically the end of my talk. I think if we do the sharing, I don't know if we're going to share slides or just the recording, but for the sake of the recording, I put together a little reading list, which you can pause just on these slides and have a little look. And these are some papers. If there's some particular things you want to read up on, you can have a look at those. But yeah, otherwise, that's the end of my little section.
All right, well, thank you very much, Rohan.
Cool.
Now we're going to hand over to Keshava. Keshava has just started at Trobe. He came from QIMR, and I think you joined us around about August, if I recall correctly.
Yep. Okay. Good afternoon. My name is Keshava Datta. I started, as Chris mentioned, at Trobe about 2.5 months back as a proteomics specialist. Before this, I was doing my postdoctoral fellowship from the Queensland Institute of Medical Research at Brisbane. Firstly, thank you, Chris and Rohan, for providing me this opportunity to present in the webinar. So let's get started. So this is an image that you will all see wherever you attend any proteomics talk, because this is the crux of any proteomics project. Essentially, we start with a protein lysate from the sample of your interest. It then either goes through a gel and the bands are excised and the gel is digested, what is called as an in-gel digestion, and the peptides are generated. Or the protein lysate is directly digested using a protease in solution and the peptides are generated. Irrespective of how the peptides were generated, these are then cleaned up to get rid of any contaminants, and then they are analyzed on the LC-MS. This data that is generated on the LC-MS is then analyzed using bioinformatics tools to make sense of it and then to interpret the data. Now, this seemingly straightforward technique can be made more complex. Different techniques can be added upon this by overlapping, say, depletion or enrichment or antibody-based pull-downs, or many other techniques can be overlapped onto this pipeline, and we can achieve a plethora of applications. Now, this is an overview of the different biological applications of proteomics. Now, please note that these categories are not set in stone. The boundaries of these categories are very fluid. So let's look at them one by one. So protein expression profiling, also commonly known as quantitative proteomics, is arguably the most commonly used technique in proteomics. Let's say you want to compare a disease state with a healthy control, or you want to compare a knockout with a wild type. What are the proteomic changes that are happening? That is where this comes into picture. The second one is post-translational modifications. I won't be exaggerating if I say that mass spectrometry has literally changed the game of the study of PTMs. Now, you could study classic PTMs such as phosphorylation or methylation, or you could also study what I would consider cool PTMs such as succinylation or crotonylation or acetylation using mass spectrometry. The third category I would consider is called functional proteomics, wherein we can identify the proteins that are interacting with the protein of your interest. And using these protein interactions, we could build protein interaction networks. Now, why do we do that? Building protein interaction networks or identifying interacting partners helps in finding out the function of a protein or in what pathways are they typically playing a role in. This is where mass spectrometry can help in. Finally, there is a term called pharmacoproteomics. I would just call it as translational proteomics because it has direct translational application. Now, let us say you have a drug that is showing you the phenotype that you want in either cell culture or mice, but you do not know what is the protein target of that particular drug. You could use proteomics to do that. Or what is even cooler is, let us say you have a protein target and you have a drug that binds to the target and is generating the phenotype of your interest. But then you want to identify what are the off-target effects of your drug. That is, what are the other proteins to which your drug is binding. That is where we come in. We can use proteomics to identify what are these off-target effects. Now, I would love to talk to you about all these techniques and all these methods and how these can be applied in research. But in the interest of time, I'll be giving one example from each of these technique categories that I have mentioned here. So first one will be a temporal analysis for protein expression profiling, phosphorylation for a PTM study, interacting partners of MYC, transcription factor, and finally drug target identification for staurosporine, a drug. The first one is quantitative proteomics for expression profiling. As I said, this is probably the most commonly used technique in proteomics. Now, there are several techniques by which we can achieve quantitative proteomics. There are labeled methods, there are label-free methods. Within labeled methods, there is metabolic labeling and chemical labeling. Within chemical labeling, if you have different companies there, they sell different kits. But I do not mean to bore you all with the technical details of all these techniques. What I mean to say from this particular flowchart is that we have a plethora of techniques in our arsenal, and we could fit your experiment into one of these based on the samples. So that leads me to the question that I typically people ask us, that what is the best quantitative proteomics method? So this intelligent fellow here says that suits your experiment the most, the one is. That is, there is no best technique for a quantitative proteomics based on the sample type and based on the number of samples and probably the budget of the project, we could decide which is the best method for your experiment. Now, as an example of quantitative proteomics, I am plugging my own study. I'm just using this to promote my own study. That was one of my projects during my postdoc. So the question that we started with was, what proteins change their expression levels in response to hypoxia in melanoma? So hypoxia, for those of you who might be interested, is less amount of oxygen. So 5% oxygen or 1% oxygen is called hypoxia. It has several physiological effects, and it is an important concept in cancer and also in many other areas of biology. So here we started with the question, as I said, what are the proteins that change their expression levels in melanoma? So what I did was I used 4 melanoma cell lines. I exposed them to increasing durations of hypoxia, 1% oxygen, starting from 0, which is the control, to 24 hours. I took all the 9 conditions and I extracted protein and I fit them into a 9-plex TMT experiment. And after that, we did peptide level fractionation using basic pH reverse phase liquid chromatography, and we collected proteomic data on a Orbitrap fusion and then analyzed the data. Just as a side note, we have an Orbitrap Eclipse here at the CPP, which is next generation of Orbitrap Fusion, so even better data. So what did I find? I found that a subset of proteins show very similar patterns of change across all 4 cell lines. And these proteins, which showed similar patterns in all 4 cell lines, I considered them as a signature. And that is pretty much the end of the proteomic data. But how did I use the proteomic data? I integrated this proteomic data with genome-wide CRISPR knockout, and then we proposed a potential therapeutic target, and then carried out functional studies to show that that particular target is indeed a good target to use in the context of hypoxia. That is about quantitative proteomics. I next move on to phosphoproteomics. Now, phosphorylation is the most commonly studied PTM. People studying cell cycle in yeast to people studying activated kinases in cancer or developing drugs against activated kinases in cancer. Everybody is interested in phosphorylation. But the issue with phosphopeptides and mass spectrometry is that phosphopeptides are stoichiometrically very low in number. That is, if I compare the number of phosphopeptides to non-phosphorylated peptides, it is a very, very low ratio. So we need to do enrichment techniques. Now, phosphorylation typically happens on serine, threonine, and tyrosine. Serine is the most phosphorylated amino acid. Threonine comes next, and tyrosine is the least number of tyrosines are phosphorylated. So if we are interested in studying either a serine or threonine phosphorylation, we carry out enrichment techniques such as immobilized metal affinity chromatography or titanium dioxide based enrichment. But if we have to do a phosphotyrosine study, we will have to use an antibody against the anti-phosphotyrosine because the phosphopeptides are that low. Once these phosphopeptides are enriched, then we can analyze it on the mass spec and then come up with a list of sites which are phosphorylated. As an application of phosphoproteomics, we will be looking at a signaling pathway by TSLP. TSLP stands for thymic stromal lymphopoietin. It's a cytokine. It plays a role in T cell activation. Essentially, what we wanted to know is the pathway that is triggered or activated when TSLP binds to its receptor, which is the TSLP receptor in combination with the IL-7R alpha. So there is a bunch of dimerization and stuff here, but anyway, the control also had receptor, but no cytokine. And then we had the cytokine treated sample as the test sample. We had built in a SciLac method here for quantification of the phosphosites. And then we extracted protein, digested it with trypsin, fractionated the sample, and then just now, as I mentioned, we carried out anti-phosphotyrosine enrichment and also titanium dioxide based enrichment because we wanted to capture both serine, threonine, and tyrosine phosphopeptides. And then using the SciLac labels, we quantified the differential phosphosites between the control and the treatment. So before we see what we identified in that study, let us see what was known in the TSLP pathway before this experiment was carried out. So this was what was known. So here you see the cytoplasm, here you see the nucleus, of a bunch of proteins here, a bunch of proteins there that were phosphorylated is what was known before we carried out this experiment. And one experiment, and this is what was known. So now this is what is considered as the activated TSLP pathway. This probably tells you gives you an example of the power of phosphoproteomics in biology. From quantitative proteomics and phosphoproteomics, now I switch gears and I move on to more qualitative proteomics, if I may call that. To identify interacting partners or protein-protein interactions, as they are called, PPIs. Now, before I start with BioID or biotin identification, I need to mention that there are other techniques to identify interacting partners through mass spectrometry. These might be called as affinity proteomics and immunoprecipitation mass spectrometry, which is APMS and IPMS. Now, if you are interested in APMS or IPMS, we can help you out there as well. But BioID is just a personal preference. So let's look at the method in BioID. So first of all, BIR-A is a biotin ligase, and it's a promiscuous biotin ligase. It's promiscuous in the sense that it biotinylates the side chain of lysine of any protein. So in this BioID method, essentially what is done is the BIR-A and the protein of interest are co-expressed in a cellular system. I just need to mention here that this method is now so popular that the BIR-A construct is commercially available. All you need to do is insert the protein of your interest here. And once you have that, that is considered as the sample. And for a control, you have a BIR-A construct only without the protein of interest. And for both the control and the test sample, we add biotin and we incubate it for a particular duration of time. And after that duration of time, we can do a harsh cell lysis. This is the major difference between IPMS and BioID. In BioID, we can do harsh lysis, and therefore we can capture more as compared to IPMS. So once we have lysed the cells and collected the lysate, then we capture the biotinylated proteins using a streptavidin affinity, and those proteins are digested using an enzyme, a protease, and then they are taken through the proteomic pipeline to identify the proteins that are in the test but not in the control. Just as an example, this is a study where they tried to identify the interacting partners of MYC. MYC is a transcription factor which is very well studied in the context of multiple cancers, and it is also known as a master regulator of metabolism. So what you see on the left-hand side is a standard BioID pipeline wherein they have carried out the exact same method that I just mentioned. But if you see the results, as I said just now, MYC is one of the most well-studied transcription factors in the context of cancer. But here, if you see the representation, there are two types of representation. One is a representation in a circle, and the other one is a square. So here, the ones that are represented in circles are all novel interacting partners that were identified in this study, as compared to squares that were already known. This is one good thing about BioID, that if we have known interacting partners, they can act as a positive control in the experiment. That means we know that the BioID experiment has indeed worked because we are capturing the known interacting partners, and it gives more confidence to the novel interacting partners that are identified. So that was about the interacting partners of MYC. From there, I move on to the last technique that I will be talking about in today's webinar. So this is thermal proteome profiling to identify drug targets. This particular method is based on a simple principle. The principle is that when you heat a protein, it precipitates out at a very particular temperature. Now, this temperature is termed as a TM, that is the melting temperature. Now, once a drug binds to a protein, the protein becomes either more stable or less stable in solution, as compared to a native protein which does not have a drug bound to it. As a result, its melting temperature changes. Now, if we could measure this difference, that is the delta Tm between the protein at the native state and the drug bound state, we could identify the potential targets. I should have probably put in the picture. Sorry about that. So to achieve that, what we do is this. You have the protein lysate. Now we have treated one with vehicle and one with the actual drug. Once you have treated the cell lysate with the vehicle and the drug, you split it into 10 parts. And each part you heat it to a different temperature. So you have 10 temperatures so that you have an entire range of melting temperatures covered. After that, you carry out a TMT-based quantitative proteomics, and you draw these curves, which are called as melt curves. This is essentially showing how much is the protein that is remaining after heating to that temperature. So if you see, this is a hypothetical figure. Between the compound and the vehicle, there is a difference in the TM, in the melting temperature. Maybe an example will be better. So here we are trying to see what are the off targets of staurosporin. Staurosporin is a drug that binds to kinases. So this is what I was mentioning. So these are the curves. The gray ones are the ones with the vehicle treated, and the whatever this color orange, the orange colored curves are for the staurosporin treated. Now, if you see the TM, the melting temperature, there is a huge difference. There is approximately a 10-degree difference between the vehicle and the staurosporin treated. That means ORCA, which is aurora kinase A, is a target. But then it is obvious it works as a positive control because it is a known target. But if you look at ferrochelatase, which is not even a kinase, it is also showing you a significant difference in temperature. So this is a novel non-specific target. While if it is a non-target, even if it is a kinase such as SLK, there will be no difference between the TM. So thermal proteome profiling can give you such interesting insights. So those were the applications for today. But if you have any other customized projects, or if you have any other technique that you would like to carry out at the platform, please drop us an email at cpp@latrobe.edu.au. I cannot promise that we will be pursuing each technique, but what I can definitely promise is that we will consider each of them with an open mind and see whether it can be carried out here. So that's it from my side today. I now leave the house open for questions. Thank you.
I'm going to thank both of our speakers today. It was certainly a very informative session, very clearly presented, and I hope that it's given our audience some ideas for their particular projects. So thank you all.